Hello everyone. In this video we are going to discuss about the next topic. So firstly, biomolecules. Inside our body, four types of biomolecules may present. First one is carbohydrates. So carbohydrates, it helps us to provide energy. So it provide energy. Next come to lipids. So lipids are the energy storage house, or we can say it store energy in the form of fats molecules. Proteins are responsible and give the structural and functional role to the body. Then the nucleic acid, fourth one is nucleic acid. It's the storehouse of information. Why we call it as storehouse? As it is having DNA and RNA, which contains the information of our inheritance. As they are the unit of inheritance, they contain the whole information of the whole information. What's the difference between RNA and DNA? DNA is having four bases like cytosine, guanine, adenine, and thymine. But the DNA, RNA, on the other hand, having cytosine, guanine, adenine, and in place of thymine, it is having uracil. So this is the main difference, that RNA is having uracil and the DNA is having thymine, and both of them are the storehouse of information, which is having all the hereditary information which is transferred from parents to their offsprings. So all the characteristics are present inside this DNA or RNA. DNA is more stable, that's why it's present in most of the eukaryotes, and RNA was the first genetic material, and it is not so stable as DNA. In RNA, at 2 prime OH, 2 prime and OH is present, which is extra OH, which is absent in DNA, which make it more unstable than DNA because it's reactive. Then discuss about the concept of central dogma. As this concept, according to this concept, DNA, which is double standard DNA, is converted or transcribed when transcriptions is taking place, transcribed into single stranded RNA, or we can say that messenger RNA. Messenger RNA, it's a single standard, and after translation it converted into proteins. So this is called as central dogma. And if the enzyme reverse transcriptase is present, it is responsible for the conversion of single stranded RNA into double stranded DNA. So this enzyme was discovered by Tamin and Baltimore. So this is responsible for the conversion of single stranded RNA into double stranded DNA. So that's why it's known as reverse transcriptase, as it reverse the transcription process, and that's why it is known as reverse transcriptase. And double stranded DNA replicates, which is called as replication process, and it replicates into double stranded DNA, makes its copy, which is then transcribed. After transcription, it's converted into translated and form proteins. Then discuss what is DNA made up of. So DNA is made up of polymer of nucleotide, and DNA is a polymer of nucleotide. It contains bases like purines and pyrimidines. Purines, adenine, guanine, and in pyrimidine, cytosine and thymine, uracil in case of RNA, sugar and phosphate group. Let's discuss about the different bonds present in the DNA structure. So this is the sugar base, pentose sugar, and pentose sugar is attached with the base by the bond which is known as glycosidic bond. So sugar and bases are joined with the glycosidic bond, and in this sugar you can see this fifth carbon. On the fifth carbon, phosphate group is attached. So when the 5 prime phosphate group attached with the OH, so here the bond is formed like this. After removal of water molecule, the bond is formed between phosphate and the 3 prime OH like this, and this bond is known as phosphodiester bond. So it's an important bond. You can see that two sugars molecules attach with each other by the help of phosphodiester bond. And you can see this, if this is a base, sugar, and on sugar base is attached, base and sugar is attached with the glycosidic bond, and there is three phosphate group like alpha, beta, and gamma. And alpha beta is removed when when it attacked to the other sugar group. But the bond between the three phosphate alpha, beta, and gamma is known as phosphoanhydride bond. So it is known as phosphoanhydride bond between these phosphate groups. But when the phosphate is attacked to other sugar OH group and form the bond, this bond is known as phosphodiester bond. Don't get confused between the phosphodiester bond and the phosphoanhydride bond. Now discuss about the DNA replication in eukaryotes and prokaryotes. So the origin of replication, we can say that usually it contains 80 rich sequence. Why 80 rich sequence? Because 80 rich sequence, 80 is having two, it's attached with two hydrogen bonds, and which is very easy to break. That's why then as compared in comparison with the guanine and cytosine, 80 rich bond can be broken easily. That's why in origin of replication, where 80 rich sequences present, from their uh, origin can be, or replication starts. So in if we talk about prokaryotes, there is ORIC, which is called as origin of replication, which is up to 245 base pair long sequence, and it is in prokaryotes single origin site is present. But in case of eukaryotes, we can see more than multiple origin sites. This is known as ARS, is autonomous replication sequence. It is of 100 base pair. Like this we can see 1 2 3 different type of different or multiple origin sites. Eukaryotes having active replication or passive replication. Active replication, for example, from one side is started origin, it started opening of the start the origin replication, and when it goes up to here, it does not need to start the replication, because from here the bubble, the application bubble goes to this, and just it helps us to open this also, and it also transcribes, replicates. This is like in case of prokaryotes, origin of replication starts from single origin. Replication is bidirectional from replication origin. As you can see, this is the origin of replication from where the bubbles opens, replication bubble opens, and like this one direction, and this is the second direction. Like this the bidirectional replication occurs in eukaryotes. But single round of replication occur because because the licensing factor which is required for the starting of the replication is degraded in S phase. That's why single round replication taking place in eukaryotes. But in case of prokaryotes, you can see this is the replication origin of replication as this, and the multiple replications start, multiple round of replications also take place in this, because because here licensing factors are not degraded in this case. That's why so prokaryote replication is also called a theta replication. Theta replication also, and multiple round of replication is going on in this prokaryotes. Some exceptions are also there. So unidirectional replications in, we can see in viruses, mitochondrial DNA replication, we see unidirectional replication, not bidirectional replication. Now let's discuss about DNA replications in is bidirectional. So J. Kerens in 1963 proved that DNA replication is semi-conservative using the technique of autoradiograph. So autoradiography, by using this technique, he proved that the DNA replication is semi-conservative. He also explained that bidirectional replication is taking place. Replication occurs in 5' to 3' direction, and it's dependent on template dependent process, and it is highly federal. Federal means it's a highly accurate. You can see this is strand, template strand 3' to 5' direction, and the leading strand where the replication is taking place is 5' to 3' direction. Here the 3' OH will attack the uh, alpha alpha phosphate group, and the beta and gamma just removed and it attached here. Like this the replication is goes on. Thrombone model, which explains the semi-discontinuous nature of replication. It shows that like this this is a fork of replication, and this is the leading strand where the replication is taking place. And like this we can see the direction of the formation of the discontinuous. You can see this is a discontinuous strands of the DNA which is synthesized like this manner. So this is thrombone model, which shows that explains that the nature of replication is semi-discontinuous, not continuous. One is continuous and other strand will form discontinuous manner it replicates. So thank you. That's all for today. Thank you for watching.